All right, in this explainer, we're going to take a deep dive into fatty acid oxidation. We'll be exploring the very distinct metabolic pathways that our bodies use to break down a whole variety of different fatty acids.
So, here's our game plan. We'll start with a quick overview, then get right into the main mitochondrial pathway. After that, we'll look at the more specialized alpha and omega oxidation routes, detail what peroxisomal beta oxidation does, and then pull it all together with a clinical focus on Zellweger syndrome.
So what's the big question we're trying to answer here? It's really this. How in the world does the body manage to break down such a diverse lineup of fatty acids? We're talking everything from your standard straight-chain molecules to much more complex branched ones.
Okay, let's kick things off with the main event, the pathway that does most of the heavy lifting, mitochondrial beta oxidation. As you can guess from the name, this all goes down in the mitochondria, and its number one job is to generate energy.
So, what exactly is beta oxidation? Well, at its core, it's a catabolic process, meaning it's all about breaking things down. The key mechanism is this. It systematically chops off 2 carbon units from a fatty acid, releasing them as acetyl-CoA. And this whole process always starts from the carboxyl end of the molecule.
Now, the details here are absolutely crucial. First, the location. This happens exclusively inside the mitochondria. Second, the process itself is incredibly precise. The fatty acid chain is always broken between the alpha-2 and the beta-3 carbons. This specific cleavage is exactly why this pathway is such a powerful energy-yielding process.
And you can really see the efficiency when you look at the output. For every single cycle of mitochondrial beta oxidation, you get 3 critical energy-rich molecules, 1 FADH2, 1 NADH, and 1 acetyl-CoA. These are the currencies that fuel other processes, like the citric acid cycle, to ultimately produce a ton of ATP.
But here's the thing. While mitochondrial beta oxidation is the workhorse, it's not a one-size-fits-all solution. For fatty acids with tricky structures, the body needs a couple of backup plans. These are the specialized alternative pathways, alpha and omega oxidation.
First up is alpha oxidation, and this one happens in the peroxisome. It has a very specific target, a branched-chain fatty acid called phytanic acid. See, phytanic acid has this 3-methyl group that completely blocks normal beta oxidation. So alpha oxidation gets around this by breaking the chain in a different spot, between the carboxyl group and the alpha-2 carbon. And it removes a single carbon as CO2. The key takeaway here? This process itself doesn't yield energy. Its job is to modify the chain so that it can then enter the beta oxidation pathway.
So what happens when this specific pathway goes wrong? That's when we see a condition called classic Ruffson's disease. It's caused by a defect in a single enzyme, phytanoyl-CoA hydroxylase. Without it, you can't perform alpha oxidation, and you get this toxic buildup of phytanic acid. Clinically, this leads to a whole host of problems like ichthyosis, retinitis pigmentosa, vision loss, peripheral neuropathy, and ataxia.
Okay, now for our third pathway, omega oxidation. This one is different again. It happens in the endoplasmic reticulum. What's really unique about it is that it targets the complete opposite end of the fatty acid, the omega carbon, which is the terminal methyl group. The end product is a dicarboxylic acid, and just like alpha oxidation, this is not an energy-yielding process.
All right, let's circle back to the peroxisome because it has another really important job to do. It has its own version of beta oxidation, and its main role is to handle very long-chain fatty acids.
Breaking down these really long-chain fatty acids, or VLCFAs, is actually a tag team effort. Step 1 happens in the peroxisome. The VLCFA goes through a sort of preliminary beta oxidation, getting chopped down until it's a medium-chain fatty acid, something like octanoyl-CoA. Super important point, this first step does not produce any energy. Then, for step 2, that shortened fatty acid is shuttled over to the mitochondria, where it can finally undergo standard beta oxidation to generate energy.
So when you put them side by side, you can really see this incredible division of labor inside the cell. Mitochondrial beta oxidation is the main power plant, breaking down standard fatty acids for energy. Peroxisomal beta oxidation, on the other hand, is like a pre-processing facility. It takes those extra large fatty acids that are too big for the mitochondria to handle, shortens them to a manageable size without making energy itself, and then sends them over to the mitochondria to finish the job.
Okay, we've covered the how. Now let's talk about the so what. What happens when these peroxisomal pathways fail? Well, the consequences are nothing short of catastrophic, and there's no better or more sobering example than Zellweger syndrome.
So, Zellweger syndrome, you might also see it called cerebrohepatorenal syndrome, is a rare inherited disease. And what makes it so devastating is that it's characterized by the complete and total absence of peroxisomes in every single tissue in the body.
Genetically, it's an autosomal recessive condition. The root of the problem is a mutation in what's called a PEX gene, which is absolutely essential for building peroxisomes in the first place. The result? A complete, generalized loss of every single function that peroxisomes are supposed to perform.
So, with no peroxisomes to do their job, you get this massive, toxic buildup of specific molecules, especially in the brain. We're talking about those very long-chain fatty acids, specifically C26 to C38 polyenoic acids. You also see an accumulation of phytanic acid, since alpha oxidation can't happen, and another molecule called pipacolic acid.
You might be wondering, what on earth is pipacolic acid? Well, it's a type of amino acid that you normally only find being processed inside peroxisomes. Because of that, when it starts accumulating in the blood and tissues, it becomes a key diagnostic marker that tells us something is very, very wrong with the peroxisomes.
And this biochemical chaos leads to a truly severe clinical picture that affects multiple body systems. We see profound neurological dysfunction like severe hypotonia and seizures, along with serious liver problems that show up as jaundice and an enlarged liver, or hepatomegaly.
1 year. That number, 1 year, is because the prognosis is, unfortunately, uniformly fatal. Because of the complete shutdown of so many critical metabolic functions, patients diagnosed with Zellweger syndrome typically die within their first year of life.